example. And transfer RNA, they are in between 10 and 15% in the cell from whole mass of all RNAs. The messenger RNA is linear and its size is variable. I think you can guess why, I already said it. And it's in the mature stage, when it is able to participate in the process of translation, it has 7-methylguanine cap and poly A tail. The messenger RNA carries the genetic code of ribosomes, means the codons that contains messenger RNA serves as a program or as matrix for ordering of the amino acids in their specific sequence in the peptide chain, in the polypeptide chain. Their synthesis happens by transcription as all other RNAs, and they are copied by the DNA sequences that are called genes. The genes contain information not only for messenger RNAs. Genes in DNA contain information also for other types of RNAs, namely ribosomal, transferred, and other heterogeneic RNAs. The ribosomal RNA is characterized with the longest between the RNAs, and when it is functional, it is associated with proteins in the content of large and small ribosomal subunits. And it is able to condense amino acids, namely because of the function of the peptidyltransferase ribozyme in the content of large subunit. The ribosomes consist of 65% of ribosomal RNAs, and the left percent are proteins. They consist of two subunits, the large one and small one, and during the lecture for translation, you'll get more details about the ribosomes. The transfer RNA. For each amino acids, there is specific transfer RNA. Why? Because each amino acid is coded by one or more codons or triplets, combinations of nucleotides in DNA sequence and or in RNA sequence, and that's why transfer RNA is specific for the amino acid. Means there are at least 30 transfer RNAs corresponding to 20 amino acids. Why they are not 20 and why sometimes they can be 61, sometimes can be 30, we'll discuss this in other lecture because time is short. And yes, this question that I'll tell you in the lecture for the genetic code. The treatment of the transfer RNA, there is the site to which the amino acid is attached. Except other loops, there are acceptor stem and site and anti-codon loop. There are two more loops that have specific function. Briefly, I'll tell you that the D arm and loop is specific for each transfer RNA, and its role or function is to recognize and attach the amino acyl synthetase, the enzyme that causes the amino acid, pairs the amino acids with the transfer RNA. The T loop or thymidine loop is non-specific. It's the same for all 30 or 60 transfer RNA because its function is to attach the transfer RNA to the ribosome. So because the ribosomes are the same, even they are many, but their content and active sites for the binding of transfer RNA is the same. That's why this T loop is the same for all the transfer RNA. It's non-specific. Except the three main types of RNA, there are several other types of RNA that are produced in the cell. In this table, they are described with abbreviation, and that means the abbreviation. Small nuclear RNAs function in a variety of nuclear processes, including the splicing of messenger RNA. The small nucleolar RNAs are used to process and actually modify ribosomal RNAs. Why nucleolar? What means nucleolus? I hope you know that this is non-membrane limited structure in the nucleus that can be seen sometimes under the electron microscope. And this is the area that is rich of ribosomal RNAs. This is the area when the ribosomal RNAs are produced and modified. That's why, except ribosomal RNAs in the nucleus, there are small nucleolar RNAs because they have a role in the processing of ribosomal RNAs that are produced at the territory of the nucleolus. Small chiral RNAs. They modify small nucleolar RNAs and small nuclear RNAs, and they are a class of small RNAs that specifically localized to the body called Cajal body. Cajal body, it's a suborganelle that is involved in the biogenesis of small nuclear ribonucleoproteins, or SNRPs. A little bit later in another lecture, we'll discuss what are SNRPs. Not Smurfs, SNRPs. Nucleus and Cajal body, we can say they are suborganelles at the territory of nucleus. MicroRNAs, miRNAs, they regulate gene expression typically by blocking translation of selective messenger RNAs. And the main difference structurally by the small interfering RNAs, which have the same function, is that miRNA, microRNAs, are single-stranded. The SI, or small interfering RNAs, are double-stranded. Guide RNAs are RNAs playing a role of modifying messenger RNA, because messenger RNA in eukaryotes usually is transcribed as non-mature pre-messenger RNA that has to be modified different ways in order to become able to participate in the translation, to serve as matrix for the protein synthesis. And except this small, generally they are short RNAs, there are other non-coding RNAs that that function in diverse cell processes, including telomere synthesis, X chromosome inactivation. Today I'll talk to you about the X chromosome inactivation, and the transport of the proteins into the endoplasmic reticulum. So this is the end of the lecture about nucleic acids. The lecture is uploaded in, I'll tell you it's in the information system. This is the page, and I have to, now I will share the, so this is the web page of the university, and information system is this one. Here, with your accounts, you can enter the system, find the discipline biology, and you'll see that you have message by me. In this message, my name is Milena Atanasova. So when you see my name and open the message, there are several messages. One of the messages contains the other contains description of the discipline. As I remember, Word file or PDF file, I don't remember how I uploaded it, doesn't matter, but it is written by description. And in the biodescription file, you'll find the description of the disciplines, the name of the assistant professors and professors in biology involved in the discipline biology. You'll see the schedule of the practices and of the lectures. You'll see literature. Some of you, colleague viewers, asked me about the literature. In this biodescription, there is a short list of literature. I will send you the same way in the information system wider or the list of literature that contains much more books. Of course, it is not normal and not necessary to find all these books I recommended to you. There is a group of books called Main, and but the main, the common between the books I recommend you that all of them are able to be found in the library, or in our library, but in the reading room, not for borrowing, because most of them are somehow taken by your colleagues from previous years and never turned back. That's why they are possible for the reading room only. So if you have some questions, some askings about the discipline, how the exam takes place, when you have lectures, when you have practices, the plans of the practical labs, etc. First, see in the description of the discipline biology. And if you cannot find the answer of your question, then ask me or my colleagues. And the other thing I sent you as message was the synopsis. And now I'll go on to the other lecture. that is organization of the genome. As you can see, in each of my PowerPoint presentation, I'm writing the topics from the synopsis that you can find in this lecture, cover the lecture. So here are they from 4th to 7th topics from the synopsis. What is genome? Definition for the genome is the haploid set of chromosomes in a gamete or microorganism, or in each cell of a multicellular organism, or the complete set of genes or genetic material present in a cell or organism. And the second definition is probably not so correct, because not all the DNA sequences in the nucleus play a role of genes, but nevertheless they are part of the genome. Because the genome includes both the genes, the coding regions, and the non-coding DNA, as well as the genetic material of the mitochondria and chloroplasts. Because in eukaryotic organisms, there is genetic material in these organelles. In the animal cells, in the mitochondria, in the plant cells, in the mitochondria and chloroplasts. You know, hope that chloroplasts are present only in plant cells and some microscopic unicellular organisms. The typical prokaryotic genome is one circular double-stranded DNA. VS is double-stranded DNA. But some prokaryotes are more exotic and except the main chromosome, this one circular double-stranded DNA, which usually is the largest one, they contain one or more smaller DNA molecules. And these smaller DNA molecules are dispensable to the life of the cell. They are called plasmids. I'm sure you heard about plasmids in prokaryotic organisms. And here are two examples. As I said, they are exotic examples because I can say the microorganisms are maybe exotic. Borrelia burgdorferi, a bacteria that causes Lyme disease in human, that is transferred by vectors as ticks. This bacteria has one large linear chromosome. Its size is 0.91 megabase pairs. MB is megabase. It could be NBP, which megabase pairs. In this case, the pairs P is omitted. And except this main chromosome, there are an additional 0.53 megabases of DNA in 17 different linear and circular molecules. Agrobacterium tumefaciens, it's Crohn's disease, this one, in some plants, contain a 3 megabases circular chromosome and 2.1 megabase linear chromosome. Prokaryotes consist of two big groups, or classified as two big groups, Eubacteria and Archaebacteria. Eu means true bacteria, Archae means old bacteria. So the old bacteria also vary in chromosomal organization, but their DNAs are only circular. And here are the two examples. Methanococcus yanachi. It's autotrophic, hyperthermophilic organism. And it has 3 chromosomes with different size. One of them is the biggest, and the other are smaller. Archaeoglobus fulgidis. It's found in high temperature oil fields. It has 2.2 megabases circular chromosome. Both bacteria groups, Eo and Archaea, lack a membrane-bounded nucleus. Hence, their classification is prokaryotes. I can understand that these are maybe your 6th hour of lectures, but it's very bad to left the lecture in the middle. If it's not interesting, tell me. Yes, I can understand you're tired, but the situation is this. I told the dean and director that it is ridiculous to have 8 hours of lecture in one day. Did you have anatomy or cytology this morning? No? So, okay, there are only 4 or 6. How many hours chemistry? Not so many. Okay, I'm going on. If in the hall there is 1 student, I'll speak. So, bacteria have no membrane-bounded nucleus. That's why they are classified as prokaryotes. Pro means till the nucleus. Karyon, nucleus. Pro means before, till the nucleus. So these are organisms that are before appearance of nucleus. The DNA of prokaryotes is densely arranged in a cytoplasmic region called nucleoid. Make difference between nucleotide and nucleoid. Nucleus-like. Oid is a suffix used in the words to say that this is something like the other structure. And the nucleolus contains all or most of the genetic material. Usually, plasmids are not in the area of the nucleoid. They are distributed in all the cell. Usually, in both prokaryotes and eukaryotes, DNA is much more longer than the size of the cell or the nucleus. That's why, in order to fit in the cell, especially in prokaryotic cell, it is necessary DNA to be somehow folded or supercoiled. And it was established in Escherichia coli, the main object for experiments with prokaryotes. It is established that the main chromosome of Escherichia coli is highly supercoiled. If the molecule of DNA is not folded, is not coiled, it will be long 1 millimeter. It's about 100 times longer than the diameter of the cell of the Escherichia coli. So DNA supercoiling helps it fit into the cell. The supercoiling happens in both directions, positive and negative. Most bacterial genomes are negatively supercoiling during normal growth. And what is positive coiling? What is negative coiling? If the DNA molecule is bound more tightly in the direction of the DNA helix, which in A and B form is right rotated, we say that the molecule is positively supercoiled. Whereas in the DNA, if the DNA is less tightly bound by applying a left-handed twisting, we say it's negatively supercoiled. The enzymes playing a role of coiling and uncoiling of DNA molecules in both prokaryotes and eukaryotes are called topoisomerases, a group of enzymes playing a role for coiling, supercoiling and uncoiling of the DNA double-stranded helices. Not only supercoiling takes place for the packaging of DNA molecules, they form additionally structures called looped domains. And each of these looped domains is supercoiling additionally. And the compaction factor for looped domains is at about 10 folds. Compaction factor means how many times decreases the length of the molecule of DNA. How many will be the looped domains in the circular molecule of the prokaryote, of DNA of the prokaryote, is species specific. Means for each species, the number of loops is specific. And it is established that in Escherichia coli, there are between 40 and 100 domains. And each of them is with size 40 kilobases. And here, how it looks like. This is uncoiled circle Escherichia coli. Is with formed looped domains that here are supercoiled, and this helps for decreasing of the diameter of the molecule. Here you can see it's 350 micrometers, here it's 30 micrometers, and here it is 2 micrometers. So the size of the prokaryotic cell is at about 10 micrometers, so 2 micrometers fitted very well in the prokaryotic cell. Except the main chromosome, there are extranuclear genetic material in prokaryotes, and its name is plasmids. They contain genes that are not obligatory for the cell, but have significance for its survival and for the competition with other cells. So one of the genes that are present in the plasmids are those for antibiotic resistance of the cell. Up to 8 genes for resisting 8 different antibiotics may be found on a single plasmid. Except these genes, there are genes for bacteriocins. These are proteinaceous toxins that are produced by the bacteria to inhibit the growth of similar or closely related bacterial strains for its competition, for surviving. There are other genes in the plasmids responsible for the pathogenicity of the bacteria because they are genes for different toxins, substances that cause illness in the host of the bacteria. And that's why different strains of bacteria can be with different pathogenic abilities. If they contain more plasmids with more toxic genes for causing the illness, they will be more pathogenic. And respectively, they'll be less pathogenic if they don't contain all the genes for all the toxins of the bacteria, or they are less as quantity. Other specific plasmid is a fertility factor. It's called F, or fertility factor, because it plays a role in conjugation. Conjugation is attachment to two bacterial cells each other with their membranes and exchange of genetic material. This much more resemble sexual process, exchange of genetic material, and because this during this conjugation the F factor is transferred between the cells, between the bacteria, that's why it's called F, fertility factor. Those that are donors of the F plasmid are F plus cells. Those accepting or receiving the F factor are recipient cells. The F factor can exist outside the bacterial chromosome, but it can be integrated into the chromosome. It's valid for all other plasmids. They can exist alone, aside from the main chromosome, but they can be integrated in the nucleoid of the bacterial cell. This was about the organization of the genome of prokaryotes. Do you have questions? Everything is clear. Okay. Eukaryotic chromosomes. Most prokaryotes consist of one chromosome. Most eukaryotes have a diploid number of chromosomes. Two copies. Why? Because of the sexual reproduction. Each chromosome in eukaryote consists, or yes, is produced by fusion of two haploid cells called during the fertilization. There is fusion of what? What's the name of the cells that are fused during fertilization? Gametes, if I heard well, gametes, and of course male and female, etc. We'll talk about this. And the genome, thank you for the people that are online. They are more than you interested. A genome is the information in one complete haploid chromosome set. And the total amount of DNA in the haploid genome of a species is its C value. This is total amount of DNA in the haploid genome, 1N. C comes from copy, because it's there is one copy. Ashvarya Devi have to or wants to ask me something. Ashvarya, I'm sorry, I'm not reading your name correctly. Ashvarya Devi, what do you want to ask me? You can write in the chat, or you can ask me by the microphone, but because you are too many people, it will be difficult. Okay, I'm going on. Maybe he do it without knowing he's doing. Okay, C value is the total amount of DNA in the haploid genome. And there is, okay, Ashvarya, I understand. There is C value paradox. What consists C value paradox? Here are shown the number of base pairs in different organisms. T4 is bacteriophage T4. HIV-1 is human immunodeficiency virus. Escherichia coli, Lilium formosanum, it's flower, etc. Zea mice, popcorn, amoeba, Drosophila, mus musculus mouse, Canis familiaris is dog, Equus caballus horse, and Homo sapiens ourselves. So here you can see the number of the nucleotide base pairs, nucleotide pairs. Please note, who of this organism has the biggest number of base pairs? Amoeba. Amoeba proteus. Hope you know what is amoeba. It's unicellular eukaryotic organisms, considered the most primitive. So I don't know if it is the most primitive. I mean, I cannot classify it as most primitive, but maybe the organisms are a little bit compound than amoeba. More compound, more complex. But so what's the value means? It's the C value paradox is that the complexity of the organism is not corresponding or is not a consequence of the size of its genome. The size can be very huge, gigantic, but this is not, the complexity is not direct consequence by this. DNA in the nucleus of eukaryotes is combined with proteins, and this complex is called chromatin. The DNA in eukaryotes is linear, double-stranded, and it is combined as much as twice proteins. And this complex is called chromatin. It is highly conserved in all eukaryotes, means it has the same content. The DNA and proteins are similar. Nevertheless, of course, there are differences, but generally it's the same. Its organization and structure. In staining of the nucleus, there are revealed two forms of chromatin, euchromatin and heterochromatin. Euchromatin condense and decondense with the cell cycle because it is actively transcribed. And usually, euchromatin lacks repetitive sequences. And it's in the active cells, it is the main part. Heterochromatin stains deeply with DNA-specific strands in stains in contrast of euchromatin. Euchromatin usually looks lighter in the nucleus. Heterochromatin is dark mass because it is throughout the whole cell cycle usually. And usually it replicates later than euchromatin and is transcriptionally inactive, means the sequences of the heterochromatins are not transcribable. They are not transcribed. And of course, not always, because it is established that heterochromatin could be divided into two groups: constitutive heterochromatin that never is transcribed and occurs at the same site in both homologous chromosomes of a pair, and consists of mostly repeated DNA, which function is any other function but not ring function. Facultative heterochromatin varies between the cell types or developmental stages of one cell or of one pair of chromosome. It contains condensed and thus inactive euchromatin. An example for such facultative heterochromatin is Barr body. Barr body is a dense, stainable structure called a Barr body because it was discovered by a scientist with family Barr. And it is established that this is one of the X chromosomes in females that is condensed. Because it's one of the X chromosome, Barr body is called also sex chromatin. This condensed X chromosome is inactive and can be observed mainly in cells, in somatic cells of females, not in males. Why? Because there is only one X chromosome, the other sexual chromosome is Y. And the Barr body can be observed in interphase, not in mitosis. In interphase, usually it is situated at the periphery of the nuclear membrane as a dark structure like this. Here it is. It's easy to observe the Barr body from the cells, from the mucosa of the internal wall of the mouth cavity. Epithelial cells stained with toluidine. Maybe you'll do this during the practices in biology classes. So we've seen something, this is the nucleus and this is the Barr body. Sometimes it is possible to see Barr body in male cells, don't worry. It's rare and usually don't think it's a kind of pathology. Sometimes it is, because there are some numerical syndromes caused by numerical mutations, like a Klinefelter syndrome, which is characterized with 47 chromosomes, 2 X and 1 Y. And generally, Klinefelters are males, but in their cells, it's possible to see Barr body because they have 2 X chromosomes. And this is one of the possibilities to diagnose the Klinefelter syndrome with cytologic instruments. During the interphase, in the cells of females, only one of the X chromosome functions. The same as in males, because they just have only one. In the females, the other copy of X chromosomes is inactive. And inactivation happens by a process called lyonization. This process is called lyonization, again, after the name of the scientist that described this process of condensation, Mary Lyon, British genetist. The bar body has a shape of drumstick. Now I'll try to increase it. Here it is, the drumstick here. Can you see? This is the bar body in the this is the bar body in the nucleus of granulocytes. Oh, in me it's seen. Now, okay, here it is. In my screen it's seen, excuse me. Now it's seen, okay? This is a granulocyte that has, its nucleus has many lobes shape, and here is this drumstick. It's called drumstick because resemble drumstick. This is the bar body sexual protein. Let me now come back. Okay, and the colleagues from the distance told me that in superfemale we can find bar body also. In superfemales, it is possible to see cells with two bar bodies because they have 47 chromosomes 3X, and the 2X, two excessive X, usually are condensed like bar bodies. Thank you. Afan. The chromatin structure. The common length of DNA in the eukaryotic cell in humans is between 1, 2, 3 meters. I met it, but it's meters. Let's us say between 1 and 2 meters. So it has to fit in 5-10 micrometers nucleus. How this happens? By packaging with help of different proteins and the way of coiling and folding. The proteins playing a role for packaging of DNA in the eukaryotic nucleus are called or are grouped into groups: histones and non-histones. The histones are a large quantity, small proteins with positively charged molecule. They are highly conserved between the species. H1 is less conservative. Non-histones are general name of other proteins. Clearly, those are not histone proteins. And they associate with DNA also. But this group consists of proteins usually are transient. And because of this, they vary widely, even in different cells from the same organism. Most of them are negatively charged and bind by attachment to the histones that are positively charged. Remember, DNA molecule is negatively charged. That's why positive and negative. Positive histones and negative DNA are attracted, and also negative non-histones are attracted by positive histones also. The amount of non-histones vary, as I said. It can be less than 100, more than all the DNA, or less than 50% of DNA mass. Depends on the stage of the development of the cell and on the type of the tissue of the cell, etc., etc. Two molecules of each histones, which are 5 different molecules, so 2 of each except H1, are associated to form a nucleosome core, or octamer. Octa, Latin 8, because 4 histones with 2 copies, so the globular structure consists of 8 subunits. That's why it's octamer. And DNA wraps around this nucleosome 1.7 times, 144 base pairs. And this wrapping decreases its size 7 times, 7-fold. And the diameter of nucleosome cores is 11 nanometers in diameter. The histones, I missed to tell you that they are named with age and numbers. H1 does not take part in the formation of nucleosome. The other, H2A, H2B, H3, and H4, they consist or are part of the nucleosome. H2A and H2B probably at the beginning were described as the same protein. That's why later, in order to be distinguished, are named this way, with A and B. And here is the nucleosome, the green one, and the DNA molecule that coils around this globular structure. And this is electronic micrograph of nucleosomes and the DNA sequences that bind the nucleosomes. And it resembles a bed on a string, a necklace bed on a string. I think it's clearly seen. The lowest level of packaging of DNA molecule in the nucleus during interphase. Here, in this schedule, you'll see the content of the nucleus, of the histones that build the nucleosomes. They are rich of lysine and arginine amino acids. Basic amino acids, and they bind to DNA mostly by salt bridge interactions to phosphates in the DNA backbone. Phosphates are negatively charged, histones are positively charged. That's why this salt bridge and attraction between them help for the binding. What's the role of H1? H1 serves as the linker histone, links the nucleosomes, and connects them to create chromatin with diameter of 30 nanometers for an additional 6-fold condensation. The linker DNA has different length, 10 to 90 base pairs, and depends on the organism. Here is the beginning of the additional condensation, again, electronic micrograph. Here is the 11 nanometer diameter of the nucleosomes, and the additional coiling thanks to the linker histone, and the formation is called solenoid. Here it is, a solenoid that has 30 nanometer fiber diameter. And the other name, except solenoid, is chromatin fibers. The stability of 30 nanometer fibers is modulated by post-translational modification of the tails of histones in the octamers, especially H4. Here again, a solenoid or chromatin fiber. This is electron micrograph, and the scheme of the coiling of this bed on a string, an additional decrease of the size of the chromatin fiber. H1 is not shown here. We can see only the green globe of nucleosome. The chromatin fiber are condensed additionally by formation of such called radial looped domains, third level of compaction. The formation of these looped domains includes interaction with the nuclear matrix. The nuclear matrix, or the other part of the nucleus except the chromatin, is composed of two parts: the nuclear lamina, the membrane, and the proteins that surrounds or contact with the membrane, and internal matrix proteins, the proteins in between the chromatin. So these proteins play a role for compaction of DNA into radial looped domains. Each loop ranges in size between 1 and 4 million base pairs in the mammals. During interphase, here are they, the looped domains, and the axis of looped domains for each chromosome is called chromosome scaffold, that is formed by non-histone proteins. Non-histone proteins. Here, the axis or scaffold made by non-histone proteins, the looped domains, and the electron micrograph of looped domains. The matrix attachment regions and scaffold attachment regions are sequences of DNA that contact with non-histone proteins. Loops are anchored to the linear matrix by these DNA sequences: Mars and SARS, matrix attachment regions, and scaffold attachment region. So these regions are sequences of DNA that interact with proteins in order to form these loops. And an average human chromosome, you know, they are with different length. So the average one contains at about 2,000 looped domains. And of course, this high level of packaging play a role in the control of the processes as transcription and replication in order to be transcribed or replicated, the DNA material has to be unpackaged. Matrix attachment regions are anchored to the nuclear matrix, thus creating radial loops. The attachment of radial loops to the nuclear matrix is important for the gene regulation and for the organization of the chromosomes within the nucleus. Each chromosome in the nucleus is located in a discrete and non-overlapping territory in the nucleus. So, observing the nucleus under the microscope, we can see a mixed mass of material, of substance, but it's not true. It's highly organized matter, and each chromosome has its own area, its own territory of localization in the nucleus. And this is proved by a technique called in situ hybridization, or interface nuclei of the interface nucleus chromosome-specific fluorescently labeled probes. And these probes indicate that chromosomes reside within restricted regions of the nucleus rather than appearing throughout the nucleus, but the precise position of the chromosomes are not reproducible between the cells. And this is a nucleus, and this image is made by computer accumulation of several other images with different fluorescence stainings. You can see that each color is different chromosome, and it has its own territory in the nucleus, its own localization. And here is the different order of chromatin packaging from the nude DNA molecule till the metaphase chromosome. Here we can see how the metaphase chromosome is depleted. I will increase the image again. So the chromosome is depleted by histones with enzymes that degrade the histone proteins. And we can see that only the DNA material left and maybe non-histone proteins. Here is the centromere, and these are sister chromatids. And here I put two web pages. The first one is animation about the organization of the DNA material. At the beginning, I tried to show this movie, but I cannot arrange the sound. There is no sound, and my colleagues from IT sector promised me to come for the next lecture, and I'll be able to show you movies with sound, because, of course, without sound, it's not good. There is information in these movies, but maybe when I turn back, I will put this lecture in the information system, so you can copy the link and to see the animation. And the other link is namely about the organization of DNA and its different level of compactization in the eukaryotic nucleus. Any questions? No? Yes? No. The structure, yes. 13 minutes left. have 13 minutes. Patient. Structure of the base chromosomes. Now, till the moment, the story was about sub-microscopic level of organization of the chromatin, because the levels of organization cannot be seen with light microscope. That's why it's sub-microscopic organization. Now, I'll tell you several words about microscopic structure. We exclude the electron microscopes. We will talking about the structure that can be seen by light microscope, and you'll see it. So the chromosomes are very well seen during metaphase of mitosis. And it has classic four-arm structure. All of you hope recognize this structure, and their classification occurs according to their size and the position of the centromere. And here are they, the three possible shapes of chromosomes in humans. Again, what happens? The computer is retarded. Excuse me, I'm going ahead. Ahoy. Reacted slowly. Now, here is the structure. Now, microscopic structure of eukaryotic chromosomes. You know them. Metacentric, submetacentric, and acrocentric. Meta means in the middle. Ancient Greek or contemporary Greek language, I cannot say exactly, but meta is in the middle. And centromere is somewhere in the middle. But in any case, the two portions of the chromosomes called arms divided by the centromere are classified as shorter and longer, P and Q. And usually, when we order chromosomes to make a karyogram, we put the short arm up and long arm down. And these are submetacentric, means the short and long arm between them, the difference is larger. And acrocentric chromosomes, usually the short arm is microscopic, barely seen. And in human individuals, there are globular structures called satellites. In the satellites, there are multiple copies of genes for ribosomal RNA. And during interphase, these genes are localized where in the nucleus? In which suborganelle? Ribosomal genes for ribosomal RNAs. Where in the nucleus there are many ribosomal RNAs. In the nucleolus. What I said when I talked about, I forgot already, about ribosomal RNAs, yes. So in the nucleolus, during interphase, are positioned these genes in the satellitic portion of acrocentric chromosomes. And here is a karyogram of human chromosomes. it consists, there are 22 pairs of autosomes and 1 pair of sex chromosomes. And they are ordered in the karyogram, in this scheme, in this schematic ordering of the metaphase chromosomes, according their length or size, according the position of the centromere, and according the presence or absence of satellites. And the groups in human karyogram are 7, A, B, C till G. And here are they. Now I'll increase this. Oh no, it's not a good idea. Or maybe it's a good idea. A group, first to third, large metacentric chromosomes. B group, fourth and fifth pairs, large submetacentric. C group is the most, with the most pairs, from 6th to 12th chromosomes, which are large metacentric and submetacentric chromosomes. D group consists of 13th, 14th, and 15th large acrocentric chromosomes. Here are they. E group are 16, 17, and 18 small metacentric chromosomes, or medium metacentric chromosomes. F, 19 and 20th pairs, which are small metacentric chromosomes, and G group are 21st and 22nd pairs, excuse me, which are acrocentric, and being acrocentric, they have satellites, as well as D group, large acrocentrics, they have satellites on their short arms. The X chromosome, the female sex chromosome, we can add to C group, because as size and position of centromere, it resembles those in the G group, and Y is resembling G group because it's small acrocentric chromosome. So what is the karyotype? The number and appearance of chromosomes in the nucleus of an eukaryotic cell. It describes the chromosome count of an organism, its length, position of the centromere, bending pattern, any differences between the sex chromosomes, and any other physical characteristics. What is bending pattern? I'll tell you a little bit later after several slides. And the preparation and study of karyotypes that are done by illustration by karyogram are part of cytogenetics and cytogenetic analysis. Why it is necessary? For identifying normal and abnormal chromosomes for clinical and research purposes. The banding is technique for staining with different techniques. Here are the different techniques here, which are described here in this web page. The link is described here, is done here, given here. And why it's called banding technique? Because the chromosomes, after staining, are with light and dark bands. And pattern of banding, of ordering of light and dark bands, depends on the chromosomes, same bands, same way. The different chromosomes from different pairs, the banding pattern is different. So the pattern of banding serves for identification of the chromosomes, serves also for identification of any changes, structural, duplications, deletions, numerical abnormalities like hyperzoomy, hypozoomy, and this is normal male karyotype, and those chromosomes that are stains darkly considered as gene poor because the dark stain material is, heterochromatin. The light part is euchromatin. It's interesting fact that, the chromosomes in the karyogram are ordered from the largest to the smallest. But it was established later when this way of ordering of the chromosome was accepted from all scientists in the world. It was established that 21st, surprisingly, is smaller than 22nd. But in order not to confuse the scientific society, it was decided to left the ordering of the chromosomes in the karyotype this way. So 21st is the smallest, not 22nd. It is established that 22nd contains twice as many genes than as 21st, at about 400, in contrast of 200 in 21st chromosome. Normal female karyotype, 2X chromosome, etc. About intelligent DNA lecture. Thank you. Free. You are free. Liberty. Thank you to my online students. Thank you.